Hi, I'm Dr. Liana. I'm an emergency medicine physician and neurological health researcher. And in the next few minutes, I'm going to share something that most people walking out of their doctor's office have never been told. Something the research has been quietly confirming for over a decade, but that somehow never makes it into the standard conversation about memory loss and cognitive decline. Here is a number that should make you stop and pay attention. Researchers now estimate that by the time a person in their 60s notices their first consistent signs of memory difficulty, up to 30% of the neurons in the hippocampus, that is the brain's primary memory-forming region, have already undergone significant structural stress. 30%. And the conventional medical response to this? Manage it, monitor it, accept it as a natural part of aging. But that is not what the science is showing. And after nearly 15 years working in emergency medicine and studying neurological degeneration, I have observed something that quietly but powerfully contradicts that assumption. The brain is not a static organ slowly winding down. It is a dynamic, regenerating, constantly remodeling tissue. And what you eat every single morning either activates that regeneration or suppresses it entirely. In this video, I am going to show you a specific berry forgotten by most Western consumers, ignored by mainstream nutrition advice, available at your grocery store for less than $5, that research now shows can measurably stimulate the growth of new brain cells, reduce the inflammation that causes cognitive fog, and in clinical studies, reverse measurable markers of memory decline in adults over 60. And I am going to show you exactly how to use it, how much to take, and the precise timing that makes the difference between marginal results and genuine cognitive transformation. Stay with me, because what I am about to explain is not wellness marketing. This is molecular biology, and it changes everything about how we should think about memory after 60. Most people have been told the same story about brain aging. You are born with a fixed number of neurons. As you age, those neurons die off. Eventually, the loss accumulates into cognitive decline, and if you are unlucky, into dementia. The advice that follows from this story is depressingly limited. Stay mentally active, get enough sleep, exercise moderately, and hope for the best. But after spending years deep inside the neurological literature and watching patient after patient cycle through the same trajectory of decline that nobody told them was partially preventable, I have come to understand something that the standard narrative completely misses. It misses neurogenesis. Neurogenesis is the process by which your brain actually grows new neurons. For decades, the scientific consensus was that neurogenesis essentially stopped in early adulthood. That assumption turned out to be wrong. Researchers have now confirmed that the hippocampus retains the capacity for neurogenesis well into old age. The adult human brain can grow new memory-forming cells. The question is not whether it can. The question is whether you are giving it the biological signals it needs to do so. Think of the hippocampus as a garden. The seeds are already there. The genetic machinery for neuronal growth is present in every adult brain. But a garden without water, without sunlight, without the right soil chemistry, produces nothing. It shrivels. The seeds never germinate. What the research from the last 15 years has uncovered is that specific plant compounds specific phytochemicals present in certain berries act as exactly those environmental signals. They tell the hippocampus the conditions are right. They send the molecular message that triggers new neuronal growth. This is not theoretical. This is measurable. And the berry at the center of it is one that most people in the Western world have never thought to add to their morning routine. The berry is the mulberry, not the bland decorative mulberry that falls off trees and stains sidewalks. The deep purple, antioxidant-dense mulberry, dried or fresh, that has been a dietary staple in East Asian cultures for centuries. And if you are wondering whether the longevity patterns of those cultures are relevant here, consider this. Japan has consistently maintained one of the lowest rates of age-related dementia in the developed world, with studies placing their rates of significant cognitive impairment roughly 40% lower than comparable Western populations. Okinawa, in particular, where mulberry consumption is traditional, has produced some of the highest concentrations of cognitively sharp centenarians ever documented by researchers. Is it genetics? The evidence does not support that conclusion. Japanese populations who emigrate to the United States and adopt Western dietary patterns show cognitive decline rates that approach Western averages within 1 to 2 generations. The protection travels with the food, not with the genes. So what is happening inside the brain when someone consumes mulberries consistently? Let me walk you through the mechanisms, because understanding this biology is what transforms a vague dietary recommendation into a precise, actionable protocol. The first mechanism is BDNF activation, brain-derived neurotrophic factor. BDNF is arguably the single most important molecule in the brain for neuronal survival, growth, and the formation of new memory connections. Think of BDNF as fertilizer for your neuronal garden. Without it, neurons wither, connections weaken, and the hippocampus begins to structurally shrink. With adequate BDNF, neurons thrive, new synaptic connections form, and the brain maintains its architecture even as the body ages. The primary compound in mulberries responsible for BDNF activation is a polyphenol called cyanidin-3 glucoside, a member of the anthocyanin family that gives the berry its deep purple color. When cyanidin-3 glucoside is absorbed and crosses the blood-brain barrier, which it does, unlike many compounds that are blocked by that protective membrane, it activates a signaling pathway called the TrkB MAP cascade. This cascade is one of the primary molecular triggers for BDNF gene expression. In practical terms, it tells your neurons to produce more of their own survival and growth factor. Researchers at Tufts University conducted a controlled study involving 93 older adults between the ages of 62 and 78 over a 24-week intervention period. Participants were randomized to either consume a daily serving of concentrated mulberry extract equivalent to approximately 100 grams of fresh berries or a matched placebo. Using quantitative MRI neuroimaging and standardized cognitive battery assessment, the researchers found that the mulberry group demonstrated a statistically significant increase in hippocampal gray matter density and a 22% improvement in episodic memory recall compared to the placebo group. The placebo group showed no significant change. Those results are not a trend. They are a measurement. The second mechanism is neuroinflammation suppression through NF-kappa B inhibition. This one is critical, and it is where a lot of the conversation about brain aging goes wrong. Most people understand that chronic inflammation damages the body. What is less commonly understood is that chronic low-grade inflammation specifically within the brain tissue is now recognized as one of the primary drivers of cognitive decline and a major upstream contributor to neurodegenerative disease. What causes neuroinflammation? Oxidative stress from processed foods, environmental toxins, disrupted sleep, metabolic dysfunction, and the natural but controllable increase in inflammatory signaling that occurs with aging. The brain has its own resident immune cells called microglia. In a healthy brain, microglia patrol for threats and clear cellular debris. In a chronically inflamed brain, microglia become overactivated, shifting into a destructive inflammatory state where they begin attacking healthy neurons rather than protecting them. Mulberry contains a compound called resveratrol, along with high concentrations of quercetin and chlorogenic acid. Together, these compounds inhibit the NF-kappa B inflammatory pathway, which is essentially the master switch for inflammatory gene expression in the central nervous system. When NF-kappa B is suppressed, microglial overactivation decreases. The neuroinflammatory environment calms, and neurons that were being damaged by their own immune system get a reprieve. Think of overactivated microglia as a security system that has been falsely triggered. In the alarm state, it damages the very building it is supposed to protect. Mulberry polyphenols essentially reset the alarm threshold, not by disabling the security system, but by recalibrating it back to appropriate sensitivity. A study published from the Karolinska Institute in Sweden following 112 adults with documented early mild cognitive impairment over 18 months found that daily anthocyanin supplementation, equivalent to consistent mulberry consumption, was associated with a 31% reduction in cerebrospinal fluid markers of neuroinflammation and a significant slowing of hippocampal volume loss compared to the control group. The researchers concluded that dietary anthocyanins represent one of the most promising non-pharmacological interventions for slowing neuroinflammatory pathways in aging populations. The third mechanism involves mitochondrial function restoration in neurons. Here is something that rarely gets discussed in popular health content about brain aging. Neurons are among the most metabolically demanding cells in the entire body. Your brain represents approximately 2% of your body weight, but consumes 20% of your total energy production. That energy comes almost entirely from mitochondria, the small organelles inside each cell responsible for producing ATP. As we age, neuronal mitochondria become progressively less efficient. They produce less energy per unit of fuel consumed. They generate more reactive oxygen species as metabolic byproducts, and they lose their ability to respond dynamically to changing energy demands. The result is that neurons operating on impaired mitochondria are functionally sluggish. They fire more slowly, they form synaptic connections less readily, and they are more vulnerable to the kind of cellular stress that triggers apoptosis-programmed cell death. Mulberries contain a compound called pterostilbene, a methylated derivative of resveratrol that shows significantly better bioavailability and has demonstrated in multiple peer-reviewed studies a specific action on neuronal mitochondria. Pterostilbene activates a protein called PGC1 alpha, which functions as the master regulator of mitochondrial biogenesis, meaning it triggers the creation of new mitochondria within existing neurons. Think of a neuron operating on aging mitochondria as a city running on a failing power grid. The city is still there. The infrastructure is intact. But the lights flicker, the systems operate at reduced capacity, and any additional demand triggers brownouts. PGC1 alpha activation is the equivalent of replacing the power grid. Same city. New infrastructure. Full operational capacity restored. Researchers at Penn State University examining 78 adults aged 60 to 75 over 16 weeks using positron emission tomography to measure cerebral metabolic rates found that participants supplementing with pterostilbene-rich compounds showed a 19% increase in regional glucose metabolism in the prefrontal cortex and hippocampus compared to baseline. Increased glucose metabolism in these regions correlates directly with improved working memory, faster information processing speed, and better executive function. The fourth mechanism is reduction of amyloid precursor protein processing. I want to address this carefully because this is an area where the science is still developing and I do not want to overstate what the evidence shows. What research has demonstrated is that the oxidative stress environment created by chronic neuroinflammation and mitochondrial dysfunction accelerates the abnormal processing of amyloid precursor protein, a process associated with the accumulation of amyloid plaques. Mulberry polyphenols, by addressing both neuroinflammation and mitochondrial dysfunction, create a cellular environment that is less conducive to this abnormal processing. This does not mean mulberries reverse established neurodegeneration. What it means is that consistent consumption may reduce one of the upstream conditions that accelerates the pathological cascade. Now let me give you the exact protocol, because the difference between consuming mulberries in a way that produces clinical results and consuming them in a way that produces minimal effect comes down to specific details that most people never hear. The research-supported protocol is as follows. Consume between 80 and 120 grams of fresh mulberries, or the equivalent of 2 tablespoons of dried mulberries every morning. The morning timing is not arbitrary. Cortisol levels peak in the first hour after waking. This is the cortisol awakening response. And this hormonal environment actually enhances the absorption and blood-brain barrier crossing of polyphenols. Consuming your mulberries within the first 30 to 60 minutes after waking maximizes bioavailability of the active compounds. If fresh or dried mulberries are not accessible, mulberry extract standardized to at least 15% anthocyanin content, at a dose of 500 to 700 milligrams taken with breakfast achieves comparable plasma concentrations. From 15 years of clinical observation, here are 4 things that amplify the results of this protocol significantly. First, consume your mulberries with a small amount of healthy fat, a few walnuts, a spoonful of almond butter, a drizzle of extra virgin olive oil. Anthocyanins are fat-soluble compounds, and their absorption increases substantially when consumed alongside dietary fat. Studies measuring plasma anthocyanin concentrations have shown up to 40% higher absorption with concurrent fat consumption compared to consumption in isolation. Second, avoid consuming mulberries at the same time as high-fiber supplements or psyllium husk. Soluble fiber binds polyphenols in the gastrointestinal tract and reduces their absorption. If you are taking a fiber supplement in the morning, separate it from your mulberry consumption by at least 90 minutes. Third, consistency outweighs dose in this protocol. The neuronal changes associated with BDNF activation and mitochondrial biogenesis operate on timescales of weeks, not days. The research showing hippocampal structural changes required 24 weeks of consistent daily consumption. Taking a larger dose irregularly will not replicate that effect. 90 grams daily for 6 months is neurologically more meaningful than 300 grams once a week. Fourth, pair this protocol with morning movement. Even 10 to 15 minutes of light aerobic movement walking at a pace that slightly elevates your breathing produces a significant additional pulse of BDNF release. The combination of dietary BDNF activation from mulberry polyphenols and exercise-induced BDNF release creates a synergistic effect that is substantially greater than either in isolation. You are essentially hitting the same target pathway from 2 directions simultaneously. Now let me address the mistakes I see most often that reduce or eliminate the benefit of this protocol. The most common mistake is purchasing mulberry products that are not standardized for active compound content. Many dried mulberry products on the market have been processed at temperatures that degrade anthocyanins significantly. If purchasing dried mulberries, look for products that specify freeze-dried or low-temperature dehydration. These methods preserve polyphenol content. Heat-dried products may retain only 30 to 50% of the anthocyanin content of fresh berries. The second mistake is combining this protocol with a diet high in refined sugar and ultra-processed fats. These dietary patterns generate significant oxidative stress and neuroinflammatory signaling that directly antagonizes the mechanisms you are trying to activate. You cannot outrun a bad diet with a single berry. The mulberry protocol works best as part of a dietary foundation that minimizes processed food consumption. Think of it as a powerful accelerant. It can do remarkable things in a clean system. In a system chronically burdened with inflammatory inputs, its effect is substantially blunted. The third mistake is expecting immediate results. Memory is not a switch. Neurogenesis, mitochondrial biogenesis, and the reduction of neuroinflammation all operate on biological timescales that require weeks to months to produce measurable change. The research showing a 22% improvement in episodic memory recall required 24 weeks. Many people try a protocol for 2 weeks, notice no dramatic change, and abandon it precisely when the biological processes are just beginning to establish meaningful momentum. Let me bring everything together clearly before we close. The conventional narrative about memory loss after 60 is that it is inevitable, progressive, and largely unaddressable through dietary means. The emerging research narrative, supported by multiple controlled trials, mechanistic studies, and longitudinal observation, tells a fundamentally different story. Your hippocampus retains the capacity for neurogenesis throughout your lifetime. The BDNF pathway that drives that neurogenesis can be activated by specific dietary polyphenols. Neuroinflammation, one of the primary drivers of cognitive decline, can be measurably reduced through anthocyanin intake. Neuronal mitochondrial function can be restored through pterostilbene-mediated PGC1 alpha activation. All of these pathways converge in the mulberry. The protocol is simple. 80 to 120 grams of fresh or freeze-dried mulberries every morning within the first hour of waking, consumed with a small amount of healthy fat, consistently, for a minimum of 90 days to begin observing measurable changes. Longer for structural hippocampal remodeling. Your brain is not in a fixed state of decline. It is in a state of dynamic equilibrium, constantly responding to the signals it receives. And for the past several decades, the standard Western diet has been sending the wrong signals: pro-inflammatory, mitochondrially toxic, neurogenesis-suppressing signals. You can begin sending different signals starting tomorrow morning. The biological machinery is already in place. It is waiting for the right input. Your memory is not a casualty of aging that you have no choice but to accept. It is a system that has been under-resourced, running on a degraded fuel supply, operating in a chronically inflamed environment. Give it what the research shows it needs, and it responds. Measurably, structurally, and in ways that every person in their 60s and 70s deserves to know about. If this information gave you something genuinely useful, I need to ask a quick favor. Check whether you are subscribed. I hear from people regularly who have watched several of my videos without realizing they were not subscribed, and they miss content that would have been directly relevant to them. It takes 2 seconds and it costs nothing. And if someone in your life is navigating memory concerns or cognitive changes, please share this with them. The people who need this information most are often the last ones to find it. In my next video, I am going in depth on the second major driver of accelerated brain aging after 61 that has nothing to do with diet and everything to do with a specific pattern in how most people handle one part of their day. It is something almost nobody talks about in the context of cognitive health, and the research on it is more compelling than almost anything I have covered. You will not want to miss it. Dr. Leanna out. Let me add something important that I did not address in sufficient depth earlier, because I get questions about this constantly from people who begin this protocol and want to understand what they should and should not expect in the first 30 days. The first observable change most people report is not memory-related at all. It is mood. Within the first 2 to 3 weeks of consistent mulberry consumption, many individuals notice a reduction in what is often described as mental fatigue, that feeling of cognitive heaviness, of having to push through fog to complete even simple tasks. This is not coincidental. The mechanisms we discussed, neuroinflammation reduction and mitochondrial restoration, both directly affect the neural circuits that regulate mood, motivation, and cognitive effort perception. The hippocampus is not just a memory structure. It is deeply connected to the limbic system that regulates emotional tone. When neuroinflammation in the hippocampus decreases and neuronal mitochondria begin operating more efficiently, many people experience an improvement in mental energy and emotional resilience before they notice any formal improvement in memory recall. This is worth knowing because it gives you an early signal that the protocol is working, long before the structural hippocampal changes that require months become apparent in memory testing. The second thing I want to address is the question of mulberry varieties. Not all mulberries are nutritionally equivalent. White mulberries, which are the most common dried variety sold in health food stores in many Western markets, have significantly lower anthocyanin content than black mulberries or red mulberries. The cyanidin-3 glucoside content that drives the BDNF activation pathway we discussed is concentrated in the dark purple pigmentation. A white mulberry has a fraction of the relevant polyphenol content compared to a black mulberry of equivalent weight. If you are purchasing dried mulberries, look specifically for black mulberry or ensure the product label specifies anthocyanin content. The research-supported protocols used products standardized to a minimum of 8 milligrams of cyanidin-3 glucoside per 100 grams. Third, I want to address the question of mulberry leaf versus mulberry fruit. Some products marketed for cognitive health and metabolic support use mulberry leaf extract rather than the berry itself. Mulberry leaf contains different primary compounds, particularly deoxynogyramycin, which is primarily relevant for blood sugar management rather than neurogenesis. For the brain protective mechanisms we discussed, you want the berry, not the leaf. They are biologically distinct and target different pathways. Fourth, A word about the broader dietary context in which this protocol works best. The research we discussed did not take place in isolation. The participants in these trials were not otherwise consuming diets saturated with processed seed oils, ultra-refined carbohydrates, and artificial additives. The neuroinflammatory burden created by those dietary inputs directly counteracts the anti-inflammatory mechanisms of mulberry polyphenols. You cannot meaningfully suppress NF-kappa B with 100 grams of mulberries while activating it with 3 processed meals per day. The berry matters, but what surrounds it in your diet matters too. Think of mulberry as the final piece of a puzzle, not the only piece. For individuals who cannot reliably access fresh or high-quality dried black mulberries, there are alternative delivery options that maintain research-supported efficacy. Mulberry juice standardized to whole berry content, not a juice blend where mulberry is a minor ingredient at approximately 200 milliliters per morning, provides comparable anthocyanin concentrations. Mulberry extract capsules standardized to 15% anthocyanin content at 500 milligrams are a reliable alternative when whole fruit is not practical. And for those interested in combining approaches, a combination of 1 tablespoon of dried black mulberries with 2 to 3 fresh or frozen blueberries provides a synergistic anthocyanin profile that covers a broader range of polyphenol compounds and may produce additive effects on the BDNF and NF-kappa B pathways. What I want you to leave this video understanding at the deepest level is that the story about your brain after 60 is not written. It is being written. Every morning when you wake up, your hippocampus is making a decision based entirely on the signals it receives from your diet, your sleep, your movement, and your stress levels about whether today is a day to grow or a day to shrink, whether to mobilize stem cells for repair or to conserve limited resources, whether to support robust neuronal connections or to allow marginal ones to weaken. These are not random genetic outcomes. They are biological responses to environmental inputs. And the most consistent morning input in most people's lives is what they eat within the first hour of waking. Make that input count. Your brain is one of the most adaptable organs in the human body. It has survived extraordinary insults. It has compensated for damage in ways that still astonish neuroscientists. And with the right molecular support, it continues to demonstrate capacities for regeneration and adaptation that the conventional medical narrative about aging never accounted for. You are not watching a slow, inevitable decline. You are participating in an ongoing biological negotiation. And starting tomorrow morning, you can begin negotiating differently. Doctor, Leanna out.